Good afternoon, everyone. Today we're talking about an unknown bacterial species that we were working on in our microbiome laboratory. My name is Alyssa Morales, and this is Cyrus Olgens, my classmate. And throughout the course of the semester, we worked on identifying an unknown bacteria that we were given at the start of the semester. Through a series of biochemical testing, we eventually were able to resolve what bacteria we were given at the beginning of the semester.
For the purpose of testing the bacteria and conducting lab experiments to observe their behavior is to better understand their identity, growth patterns, and how they respond to different conditions. By identifying the type of bacteria present, scientists can determine whether it's harmful or beneficial and which treatments or antibiotics are most effective. Lab experiments also help reveal how bacteria spread, survive, and react to changes in their environment, which is essential for preventing infections, developing vaccines, and improving overall public health and safety.
So at the beginning of our class, we were instructed to create something that's called a dichotomous key. A dichotomous key is kind of like a flow chart, and it helps you move along from the start of zero information on a bacteria and eventually through a series of testing. If you follow this flow chart with your answers to every test, you'll eventually arrive at a possibility of a bacteria that you can then confirm with retesting. This is the dichotomous key for our group, and this is an example here in the green highlight. You can see the different tests that we decided to test our bacteria with. And as you move along, eventually all the way in the bottom row, you will see that there will be several bacteria that can only correspond to these specific answers. Um, and so this is an image of me and Cyrus's dichotomous key.
Why do we use biochemical testing? The reason is biochemical tests are used to identify unknown bacteria by analyzing their metabolic and chemical characteristics. Different bacterial species produce unique enzymes and react differently to specific substrates. These reactions help distinguish one type of bacteria from another based on patterns of biochemical activity.
Are biochemical tests helpful in detecting bacterial species? Given in our class, they were very helpful. We were able to run biochemical tests multiple times, just to reconfirm our answers. And most importantly, they're helpful because they provide reliable and specific information about the identity of a bacteria. By detecting specific enzyme activities, fermentation abilities, or other metabolic traits, these tests help microbiologists sort of pinpoint a specific species present. This is what we were attempting to do in our class, and we successfully, as you'll see at the end of this presentation, we did. And ultimately, in the grand scheme, this is essential for diagnosing infections, determining treatment options, and conducting research.
The biochemical tests we ran were the gram stain, phenol red glucose fermentation, phenol red lactose fermentation, Kia test, and oxidate tests.
So these are our results. This was our culture, our bacterial culture at the beginning of the course, with our unknown bacteria, and then using parts of this culture, so using excreting some of this bacterial colony, we would test in the second photo our glucose test, our lactose fermentation test, our Kia test on this slant, and ultimately our oxidase tests. And in the upcoming slides, you'll see what these results mean.
Interpretation of results. The bacterium has a thin peptoglycan layer in its cell wall and does not retain the crystal violet stain. This is typical for gram-negative bacteria. The bacterium can ferment glucose-producing acid, which lowers the pH and turns the medium yellow. This means it can metabolize glucose. The bacterium cannot ferment lactose, so it does not produce acid when lactose is provided in the medium. The bacterium does not ferment either glucose or lactose, nor does it produce gas or hydrogen sulfide. The bacterium does not produce the oxidase enzyme, which helps it differentiate between bacteria like Pseudomonas and others like Enterobacteriaceae.
And so the conclusion of our results, so our gram-negative testing showed that it was a Shigella species, including Shigella flexneri, which is a gram-negative bacteria. The glucose fermentation positive result showed us that flexneri can ferment glucose, which is consistent with our test results. Lactose fermentation negative, flexneri does not ferment lactose, which is typical for the Shigella species. The Kia test was negative, which shows that it does not ferment glucose in the Kia test and it does not produce gas or hydrogen sulfide. The oxidase test was negative, which was also an expected result. And this is kind of showing an AI representation and conclusion as far as how we were able to use AI to help us in our investigations of this bacteria. And ultimately, the AI conclusions went along the lines of what we actually tested in the lab. And so this led us to believe that AI can make identifying bacteria a lot easier, faster, and more accurate by analyzing data, recognizing patterns, and comparing it to already known information. And it can even predict future antibiotic resistance. And so this is why the rise of AI in educational institutions is becoming somewhat helpful, because it can speed up the processes that can take hours to weeks in the lab. And so this is an image of our confirmed and concluded dichotomous key. So as you see, like we said, our gram stain was negative. Our then phenyl glucose fermentation test was positive, positive for gas, positive for acid. And then our lactose fermentation negative. And then our Kia test negative, which led us to an oxidase test, which would help us differentiate between Pseudomonas aeruginosa and Shigella flexneri. And so that test ended up being negative, which ultimately led to our discovery of our unknown bacteria being Shigella flexneri.
As Alyssa said earlier, or unknown with Shigella flexneri. Shigella flexneri causes shigellosis, which leads to severe diarrhea, often with blood or mucus, stomach cramps, fever, and sometimes vomiting. It spreads through contaminated food, water, or direct contact with infected people. It can be mild or severe, and especially risky for young children, older adults, or other people with weak immune systems. Good hygiene, like washing hands, help prevent the spread. Antibiotics can treat the infection as well. Thank you. This was all for our project.
And we hope you enjoyed our presentation. Thank you so much.